All right, so we're going to start with chapter 3 right now, and this is bioenergetics. So we're going to look at how we get the energy not only during just physical activity, but also just in our daily lives every day, how we get the energy that is needed to contract the muscles and do all the processes necessary within the body. All right, so how we control the rate of this energy production, there's a few different ways. First off, the availability of the actual substrate itself. And the substrate we're talking about, or these substrates, would be like carbohydrates, fatty acids, protein. We'll get into more specifics about those here in a little while. But the more than we have available, the more we tend to use of those. If we end up running out of a lot of carbohydrates in the body only has a certain amount of stores. So if we end up running out of them, then we're going to start to use something else or use more of something else. And it also depends sometimes on how intense the activity is. And we'll see that later on as well. Now, also the enzyme activity. Again, an enzyme really is just a product that helps decrease the activation energy that is necessary for a reaction to occur. So if the enzymes weren't there, you would need a lot more energy to take one form of a compound and create it into something else, right, throughout a chemical reaction. So the enzymes are very important in this. And in fact, we actually have rate limiting enzymes during these processes that really facilitate how the reactions are basically regulated. The more of the end product that we get, these rate limiting enzymes will start to slow down. The less of that product that we want to make that is available, then we're going to speed that reaction up. And that is due to the activation of the rate limiting enzymes. So, and this is also influenced by a negative feedback system. All right, so a couple figures in your book that you do need to be aware of is that not only is maybe the substrates and the substrate availability important, but again, going back to the enzymes are really important in all of these processes. And a couple of things will affect the enzyme activity. One of them being body temperature. If the body temperature is too cold, the internal core body temperature is too cold, it will start to affect the enzyme activity. If it's too warm, it also affects it as well. And from these two figures, you can see that temperature, and this is in degrees Celsius, there's kind of I guess you could look at it as a Goldilocks portion there, where it's not too cold, it's not too hot, it's just right for maximal enzyme activity. So we're looking at about 37 degrees Celsius to about 40 degrees Celsius. If your body gets colder than that, the enzyme activity starts to slow down. If it gets way above 40 degrees Celsius, it starts to rapidly slow down. Now, the same thing can happen with pH. The lower the pH, or the more acidic the blood gets, the enzyme activity tends to decrease pretty dramatically. And in fact, if we get too low, some of those enzymes almost cease to even do what they're supposed to do. Now, if it gets too high, if the blood becomes too basic, that can also decrease the enzyme activity as well. Now, what I was talking about earlier about the negative feedback is that we have a product that we're trying to create. If we're creating a lot of it, that means this product is increasing. That feeds back to decrease the activity of this rate limiting enzyme. So as this increases, the enzyme activity decreases, and that's what a negative feedback is. It's one is increasing, the other one is decreasing. If the end product, if there's not a lot there and we need it, so there's less of it, there's a decrease in it, it will increase the enzyme activity. All right, so let's look at how we get energy to start off with here. First off, pretty much everything is derived from the sun. That 91 to 93 million mile travel that the sunlight ventures to get to us. It usually takes 7 or 8 minutes or so. That sunlight is important because it helps the plants grow. And also with the addition of water and various other nutrients in the soils. But because of that sunlight, the plants go through photosynthesis, they grow, and we can eat the plants then. Here's our humans over here. I know this is kind of a crude figure, but we will eat the plants. And from those, we can get carbohydrates, fats. There are some fats in plants. And usually the plants have the good fats, the stuff that we need, as you can look at as kind of the healthy fats. We can also get vitamins, minerals. There's some protein in some of the plants as well. Now, animals can also eat the plants, and we can benefit from that as well from eating the animals. So from the animals, obviously, we're going to get some protein because of the muscle tissue. We'll also get fats. There's some vitamins and minerals in there also in meat. And this isn't necessarily just animals. It could also be fish as well. Now, the amino acids that we get from eating meat are very important as well. There are some amino acids that we cannot produce ourselves in our body that we need to get from the outside. And animal foods, basically meat, contains all of those amino acids that we need. Plants are more of an incomplete protein source, meaning that if you are vegetarian, if you choose or you can't eat meat or animal products, then you will have to be careful of what plants you eat because you need to get a certain number of amino acids in you that we cannot actually produce in our own bodies. All right, so energy. Energy is basically just heat production. Usually in our bodies, most of the energy that we're creating is actually in the form of heat. That's why our bodies warm up as we exercise, as we move around. And it's also why when we pass away, our bodies get cold. There's not a lot of activity going on inside our bodies to keep it warm. So anyway, getting away from that, that's kind of a sad topic. But anyway, just kind of what happens when we pass away, I guess. Anyway, the calories that we all love and know. It's kind of a double-edged sword, calories, because calories are very, very important. That's the whole point of eating food, really, is to get calories, get those nutrients, those caloric foods in our bodies. So 1 calorie, 1 small c calorie, is equal to the heat that is required, or the energy that is required to raise 1 gram of water 1 degree Celsius. Now that small c calorie is also known as a scientific calorie. It is actually 1,000th of a kilocalorie, which is a big C calorie, and that is what we actually look at when we look at the calorie count on a food label. That big C calorie. All right, so that big C calorie is what we actually see on a food label. Right, so that's what we're talking about when we talk about calories normally. If you're a chemist, a lot of times you'll talk about the small c calories. But nutrition and exercise physiology, exercise science, most of the time we're discussing the kilocalories or the kcals. So really, if you look at a bag of potato chips, maybe has 200 calories in that whole bag, it's not really 200 small c calories, it's 200,000 small c calories. So that's the difference there between those, the different classifications. All right, so the fuel for exercise. Kind of discussed at least 3 of these prior here: carbohydrates, fats, and protein. And you can see the abbreviations there. Carbohydrates a lot of times is abbreviated capital CHO, and fats, you will see it as FA or FFA, basically meaning free fatty acids or fatty acids. And then you have protein. So those are the three big ones. And as you can see at the bottom here, there is an additional one, which is alcohol, 7 kcal per gram. But that is normally seen as empty calories, just for the simple fact that a lot of things such as beer and maybe hard liquor, they don't necessarily provide a lot of other stuff in there, a lot of vitamins, minerals. Obviously, if you have some mixed drinks, that could maybe change some things. But generally speaking, things that have alcohol, especially a high alcohol content, don't necessarily provide a lot of nutritious value. So anyway, going back to the carbohydrates, fat, and protein, all of them contain carbon, hydrogen, oxygen, and protein is a little bit different in that it has nitrogen in it. And that can be a little problematic for the body because the body does need to get rid of that nitrogen when it breaks down protein and amino acids for energy. And we end up excreting that through our urine then. And in fact, to get rid of that nitrogen, we actually expend some calories. We use up some ATP, some high energy phosphate. And we'll discuss the ATP here in a second, but we end up using some of the calories just to get rid of that nitrogen. So if we look at protein, 4.1 kcals per gram, it's the same as carbohydrates. However, protein generally, you'll see a lot of sources where protein is actually about 5.5 kcals per gram, but we end up using almost 1.5 kcals just to get rid of the nitrogen. So we only yield approximately 4.1. Now, fats, on the other hand, they provide a lot of calories, which make them really, really good for storage. That's why we store fat in large amounts. And, our bodies really tap into that in times of starvation or famine. So from an evolutionary standpoint, for the way our bodies are made, that's just really the best way to get a lot of calories in a small sample, basically. So just a small gram has 9.4 kcals, which is pretty significant. So really nutrient, or at least calorically dense, are fatty acids. Now the fatty acids are going to be stored as what's known as triglycerides, or you'll also see it as triacylglycerides, and they're stored in the adipose tissue. Now the protein is going to be stored obviously in the muscles, and carbohydrates can also be stored. Carbohydrates are stored in both the liver and the muscle, and they are stored. The storage form of that is glycogen then. Now, another thing that I do like to point out is that we don't convert energy, even though that term is used a lot of times. We don't convert anything into energy or pure energy, because that would be the E=mc² deal with Einstein, and we'd be walking nuclear warheads basically. So all we do is take those bonds in those structures, we cleave those bonds, and then we're able to transfer that energy into one form to another. All right, so let's look at carbohydrates first. So if we bring in some carbohydrates, we eat something. It's first converted to glucose, and we can use it a couple of different ways. We can use it for ATP in the muscles and the brain. The brain only runs on glucose. Well, I want to say it prefers glucose. There's another form known as ketone bodies that the brain can run on as well. But that's only in times when we don't have any glucose available. And ketones is basically a skeleton structure of glucose, essentially. So the brain really needs to function off glucose. That's why it's important when you wake up in the morning to get some type of either bread or something with carbs in it, with glucose in it, because that helps jumpstart the brain in the morning. In fact, there's a lot of research that has gone into brain activity and attentiveness and memory and just attention spans from people who eat breakfast versus not eating breakfast. So again, the storage form of carbohydrates then is going to be glycogen. And again, this is in the muscles and the liver. And when we need the energy necessary, we're going to break that glycogen down and, we're going to use it to get some ATP. However, the stores are limited. We only have anywhere from 2,000 to 2,500 calories from this, even though that might sound like a lot. And normally that's enough to get us through the day if we don't have any food coming in. If we're starving ourselves or something's happening where we can't eat or we don't have any food, whatever the case may be. But the stores are limited. If you run a marathon and you don't bring anything into your body, eventually you're going to run out of the stored glycogen and you're going to have to start relying on fats predominantly. All right, so looking at fat, it is a very efficient storage source of calories. Most of the time, this is going to be used when we start to run out, at least predominantly used when we start to run out of carbohydrates in our body, stored carbohydrates. Or it can also be used, and in fact, right now, as you're sitting, if you're watching this video, or if you're listening somehow, or you're sitting in class, if you're resting, more than likely you're probably at either burning 50% fats or 50% carbs, or you're actually a little bit higher on the fat side of things. You're probably burning a little bit more fat than you are carbohydrates. And the lower the intensity of the exercise is, the more fats you'll be burning. The higher the intensity, the more carbohydrates you're burning, because that's usually a quick energy source, as well as it's more efficient. And we'll see examples of that here later on. So again, it has a high net ATP, but again, it is a little bit slower. It's not as efficient at getting that ATP. And again, the storage form is triglycerides or triglycerides, and they're stored in the adipose tissue, the subcutaneous adipose tissue. And it has to be broken up into fatty acids, the free fatty acids, and glycerol. And that's what the two components are basically, is you have 3 fatty acids, which is where the tri comes from, and you have a glycerol molecule. And we'll break that apart right at the start of exercise, and if it's not used, it's just going to re-estrify. It's going to combine back up together, and we can store it again for a later time when we actually do need it. Protein. Protein is usually not used predominantly for energy production unless, again, it's a period of severe starvation or certain diets, possibly very low carbohydrate diets like Atkins diet and whatnot. You're probably going to be burning a little bit more protein than normal, but it's not a very good source to rely on for energy, for ATP, because it's going to take a little bit longer. If you've ever been on a low carbohydrate diet, you probably have felt that feeling of being really tired and just felt like you had no energy at all. And in fact, that's one of the severe side effects, really, of a low carbohydrate diet. And it's really not recommended for anyone who is partaking any type of athletic sport or any physical activity, strenuous exercise activity, or even working condition. If you're in, if your work requires some type of labor and you need to have as much strength and you do need to have as much energy as possible, a low carbohydrate diet is not really the way to go with that. You really need to have some type of carbs. You need to get some sugar in you. So the protein, we're going to break it down into amino acids, and it's going to be stored or it can be used. Now, those amino acids, need to first be converted into something that we can use. And normally it's going to be either converted into one of the compounds within the system. And we'll see those systems here in a little bit. Those systems that break down carbohydrates, or it's going to be made into a carbohydrate, glucose, through a process known as gluconeogenesis. So even if we don't have carbohydrates, the body's going to try to either make carbohydrates itself, or it's going to break down the protein of the amino acids and form a glucose or carbohydrate-like structure so that we can actually use it. They can also be converted into free fatty acids through a process known as lipogenesis, so creating fats. So if we get too much protein, it can be converted into fatty acids. All right, so this is the overview. This is a really rough overview of metabolism. All right, I know it looks confusing, but we'll go through it here really quickly. So this figure is not in your book, but I really like using this figure because it is a really nice overview of what's going on when we eat something and how the different sources of energy react and what we can do with each of them. So we bring in some food. All right, say we eat a nice big old steak. And we got some protein in there. And we're going to break that protein down into amino acids. And those amino acids are going to be transported in the blood and either go a couple different ways. They can go to body protein, which is basically building up the muscle tissue. And from there, or instead of the body protein, they could go into the amino acid pool. From the amino acid pool, from here, they can basically do a lot of stuff. They can go either into metabolism, or they can just be broken down. They need to be broken down. They can be created into glucose through gluconeogenesis. Again, gluconeogenesis, genesis, like in the beginning. So you're creating glucose. And in fact, it's creating new glucose, neo being new. So the amino acids can be created into glucose, go into metabolism, or from here, they can actually be made into fatty acids, into fatty acids as well. Go right into the fatty acid pool. In fact, that's not necessarily in here, but there's a roundabout way that they will be. Basically through glucose, then it's going to be converted into fatty acids. Now, from the body protein, we can break that protein down. Or if it's in the amino acid pool and we need more protein in the body to build the muscle tissue, then it's going to go into protein synthesis to build that tissue up. Now let's look at carbohydrates. So those carbs are going to be broken down and we got some glucose. Any excess, we're going to store it as fat through lipogenesis, so creating fat. If we don't create fat, we don't have excess, we're going to either store it into the glycogen stores, again, either in the liver or the muscle, or they'll go into the glucose pool. And this pool can be tapped into and we can use it for immediate energy, for metabolism, or if we don't use it, we can store it through glycogenesis. Glycogenesis is the formation, the creation, the buildup of glycogen. And then glycogenolysis is breaking down the glycogen. So these terms, glycogenolysis, glycogenesis, gluconeogenesis, remember those. Know the definitions of those. Also lipogenesis as well. Now, on the other side of this, we got fats or triglycerides, triacylglycerides as well. The other term for it. So we're going to break these down into free fatty acids. From here, then go into the free fatty acid pool or the fat stores. The fat stores obviously are going to be used for energy for a later period in time. And when we need that ATP from that fat, we can break it down through a process known as lipolysis or lipolysis, tomato, however you would like to say it. Now, the free fatty acid pool, if we're not using them, we can just transfer that and create some fat through lipogenesis. So creating fat stores. All right. So the energy systems. All right. So the three big ones that we're going to be looking at here, and these are fundamental to understanding what occurs during exercise. And again, these just don't happen just during exercise. They occur 24 hours a day, 7 days a week, 365 days a year, or 66, depending on what year it is, I guess. So they're occurring all the time. So the first one, actually the first two are going to be anaerobic. The first one is ATP PCR system. This is purely anaerobic, and anaerobic meaning that there is no oxygen that is needed for these systems to run. The ATP PCR system we'll get into shortly. It is also known as the phosphagen system. The second one, we have glycolytic system. This is also anaerobic. This involves a process known as glycolysis. And in fact, this is also referred to as anaerobic glycolysis. Now, the third one is oxidative system, or the aerobic system. And this is where we're going to need oxygen for this to run. All right, the ATP PCR system, the very first one here. So it stands for adenosine triphosphate dash creatine phosphate, or phosphocreatine. So in the muscles, we're going to be breaking apart ATP, and we're going to be creating ADP and inorganic phosphate. Just remind you, if you don't remember what the ATP, ADP, all that stands for, refer to the skeletal muscle function within the textbook. But adenosine triphosphate has an adenosine, and then there's three inorganic phosphates, or three phosphate molecules attached to it. When we break it apart during muscular contraction, we are left with adenosine diphosphate. That's where the D comes from, in one of those inorganic phosphates. So then we don't convert, we transfer that energy into some form that we can use. And in this case, it'll be mechanical energy, so we can contract the muscles. When we do this, then our ATP levels decrease. We have a very finite, very small amount of stored ATP within the muscles. It's going to be eliminated fairly quickly within the first few seconds of muscular contraction, of heavy, forceful muscular contraction. This system is really going to help replenish the ATP stores by adding back on the inorganic phosphate, because it's basically donating a phosphate. And as you can see, this word right here, creatine, that is where the supplement comes from. The creatine supplementation comes from. So the theory is the more creatine you have, the more ATP that you can start to create within the body, especially during anaerobic type of exercise. That means you can possibly go a little bit longer in that exercise without fatiguing. So it should help you lift a little bit more weight, maybe at least get a few more reps. And hopefully that translates into increasing either muscular strength, power, or endurance. And you can look at some of the research on that. Some of it says yes, some says no. Most of it really says it's really not all that harmful for the most part. And it's kind of a thing, as long as you don't have any underlying condition, you're probably okay to take it. And there's probably some benefit to it. But that's going to be up to you whether you want to take that or not. And also, there's also some governing bodies that I believe that do try to limit the use of it. But it is important for this reaction. And we get it naturally. We get it from some of the foods that we eat. All right, again, so the muscular contractions at the beginning of exercise, they're going to start using up the ATP. And within the first few seconds, this system is going to start producing or start giving up the inorganic phosphate to add back on to the ADP. All right, so this is what we're talking about here. We have this phosphocreatine molecule or compound. And we're going to break it apart. So we're left with creatine and one inorganic phosphate. Now, the enzyme necessary for this reaction to occur is known as creatine kinase. This is the rate-limiting enzyme for this reaction. Know the rate-limiting enzyme. Now, when we split it apart, there's going to be some energy that is released from that. That energy is actually used to put back on this inorganic phosphate that we cleaved from the creatine, and we're able to add it on to an ADP molecule. Again, these ADP molecules are coming from muscular contraction because we're splitting up the ATP. So we have the inorganic phosphate being added on to ADP, adenosine diphosphate, 2 phosphate molecules, and now we're adding back on 1. So we end up again with a new, fresh adenosine triphosphate. Now, looking at the rate-limiting enzyme for this reaction, creatine kinase, again, it catalyzes the breakdown of phosphocreatine. It helps it go, basically. And it controls the rate of the ATP production through this pathway. Again, it's a negative feedback. So if we are exercising for contracting our muscles, we're starting to break apart the ATP during that muscular contraction. So our ATP levels within the muscles are starting to go down. That translates into an increase in ADP. We also have a bunch of inorganic phosphate floating around as well. But we're using up that ATP, and that ADP is on the rise. So this decrease in the ATP causes an increase in the activity of creatine kinase. If we stopped exercising and we slowed down the use of ATP, we're going to have an increase in the concentration of ATP within the muscles. And that will cause a decrease in the ADP because now we're not breaking apart. We're not splitting apart ATP. So we have a decrease in the ADP. So that causes a decrease in the activity of creatine kinase. So again, negative feedback. One goes up, the other one goes down. This one, where we're exercising, we have a decrease in ATP, so we have an increase in creatine kinase. All right, so the second one we're going to look at, the second system, is the glycolytic system. And again, I mentioned this before, but it is known as also as anaerobic glycolysis. And in fact, the same pathway that this is, which is glycolysis, is going to be the same pathway that we start off with when we go through the aerobic system as well. So this is going to involve an incomplete breakdown of glucose into lactic acid. And lactic acid, and you also see the term lactate. They both, in general, and this is kind of bad to do, but they're both used interchangeably, although it's generally not good to do it because lactic acid is converted almost immediately within the body into lactate. And with that conversion, there's a hydrogen ion that is associated with that. And that hydrogen ion is important because that can decrease the pH of the muscle, or basically the pH within the muscle and also within the blood, causing everything to become a little bit more acidic, which then translates into a little bit more fatigue. So the more lactate you're producing, the more hydrogen ions that you have, which will decrease the pH of the muscle, which can be problematic. So from this, we're either going to start from glucose, which is from blood glucose, which is BG, or we're actually going to get the glucose from glycogen. Now, we can generate the ATP really quickly, and we can produce quite a bit of it, relatively speaking, but it's not a lot. It's not a huge, massive amount, but we can generate it really quickly, and this is for high, intense exercise. Basically, starting off at about 25 to 30 seconds up to about 3 minutes is where this system is going to provide much of the ATP that's necessary. Now, you don't necessarily need to memorize the ATP yield. This is here just for a reference, just to show how efficient some of these are, or these three systems are. So for glycogen, if we use glycogen and break it down, for every unit of glycogen, either 1 mole, you look at one compound, one molecule, we could look at 1 gram, doesn't really matter. We're going to get 3 ATP. For glucose, we end up using an ATP right off the bat just to break it down in the first step. So we end up with a 2-to-1 ratio with that, and we'll see here in a second. All right, so this is glycolysis, and you can see the first step when we start off with glucose, we're going to use an ATP right off the bat. So you don't need to memorize these reactions. You don't need to memorize the intermediates of these reactions like glucose 6-phosphate, fructose 6-phosphate, all that stuff. You don't necessarily have to remember that. I want you to know what the pathways do, basically. Kind of be able to translate what they provide when they provide it. In this case, this anaerobic glycolysis will provide some ATP during some type of anaerobic exercise, lasting approximately 25 seconds to approximately 3 minutes in length. Now, also this step right here, where fructose 6-phosphate is being converted into fructose 1,6-bisphosphate, this is where the rate-limiting enzyme for glycolysis is located, and this is known as phosphofructokinase, or also known as PFK. We'll get into that shortly as well. So from that point of this point, right past the rate-limiting enzyme step, we're actually going to start, this fructose 1,6-bisphosphate ends up splitting up into 2. And from then on, we're going to end up with 2 separate compounds on each side. And in fact, when we get to, and this is kind of important too, when we get to aerobic metabolism, this is going to be 2 separate pathways. All right. So whatever we do on this side, we're also doing the same thing on this side. So that's beneficial for producing ATP. So not only do we just get 1 ATP that's created in this step, we actually get 2 because we have 2 separate pathways because it's split right here. All right, so you can see that this is pyruvic acid to lactic acid. Again, this is basically the last step of anaerobic glycolysis. We're going from, let me go back a slide here. We're going from this pyruvate. Again, this is going to be 2 times. So you're going to multiply this by 2, but this is the step on both sides of that. Basically pyruvate to lactate. Let's look at it kind of overall. Again, this is a figure not in your textbook, but this is another more of a simplification, I guess, in another textbook, but I will be providing these online as well. You can see here we got 2 pyruvate, and we have the lactic acid on either side. So again, each of those pyruvate are being converted into lactic acid. Now, if you look, the arrows There's two arrows, and they can go either way. In the presence of oxygen, and generally this is going to be in type 1 muscle fibers, the fibers that are more aerobic in nature, they tend to have an isoform or a certain form of the enzyme that's necessary in this step for pyruvate to lactate. It is known as lactate dehydrogenase. They have an isoform that promotes lactate to pyruvate. All right, so that's important, because if it was anaerobic, the lactate is going to be the last step. If it's aerobic, we're going to continue this reaction. We'll see it here in a little bit with the aerobic system. If it's aerobic, it's going to continue on downward, and we're going to end up getting into what's known as the citric acid cycle. Now, in type 2 muscle fibers, the ones that are used predominantly for anaerobic exercise, lifting, sprinting, those types of activities, the form of the enzyme in this case promotes pyruvate to lactate, right? So, which is beneficial because the type 2 muscle fibers are generally not needed and not used a whole lot for aerobic exercise. All right, so let's look at lactate a little bit here. It's kind of gotten a bad rap. Lactate isn't necessarily all that bad. There's actually some benefit for it. First off, we can actually use it for energy. You can convert it back into pyruvate and go right into the aerobic system and use it for energy. It can also be transported back to the liver, and we can go through gluconeogenesis, creating new glucose, and we can then use it for energy, or we can store it for glycogen. So it's kind of a win-win. We're kind of recycling some of the waste that we have. I mean, if you talk about going green, our body is the first going green. I don't know, that kind of sounded weird, but I'll leave it in this presentation. Anyway, we're going green anytime we're doing some type of anaerobic exercise, just because of lactate. I'm going to see how many people catch that in the video and see if I get any emails from it. If you listen to this, you need to email me, go green. No, please don't. Okay, so we're going to keep on going here. All right, so that lactate then, again, there's going to be some hydrogen ions that are associated with that. They're going to be created because of that lactic acid to lactate. And that is what causes the decrease in the pH, and that will cause some issues and cause the fatigue. Now, the phosphofructokinase, that rate-limiting enzyme in this step, can act the same way as creatine kinase did in the ATP-PCR system, in that there's a decrease in ATP. There'll be an increase in ADP. Because we're exercising, we are moving the muscles, we're breaking apart the ATP. That increases the PFK activity so that we can get more ATP. Now, if we stopped exercising, stop moving the muscles, we're not breaking apart the ATP, we're going to decrease the activity of PFK. A little bit more about lactate. So if the production of lactate exceeds the clearance, so we're not able to clear it out in a sufficient manner, it's going to accumulate, and the pH of the blood is going to decrease significantly, and that's what's going to cause fatigue, and the fatigue will be pretty dramatic. This has a couple of different names. Sometimes it's referred to as hitting the wall, more of an anaerobic hitting the wall. There's also a hitting the wall aerobically. It's also known as lactate threshold. So it is this point in time where the increase in lactate, or the increase in lactate significantly increases versus the clearance rate. Again, it can be problematic. Now, one way to counteract this is getting some bicarbonate supplementation in you. And if you'd like to read up on that, there's plenty of sources on that. All right, the glycolytic system, again, this is going to be a main supplier only after the ATP and only after the ATP is not being able to be supplied by the PCR system. So it's kind of a backup for high intensity exercise. We get to, 20, 30 seconds of basically sprinting, and you kind of hit that point where you can't really go on anymore unless you have a backup. Now, you can't maintain that same intensity for very much longer, past 20 seconds or so, because you're not able to generate enough ATP quickly enough. But you're able to do it pretty good up to about, again, 2.5 to 3 minutes. All right, so let's move on to the oxidative system, also known as the aerobic system. All right, this is going to involve some pathways here. Glycolysis, we've already seen glycolysis. We also have the Krebs cycle. This is also known as a couple different things. TCA stands for tricarboxylic acid cycle, and CAC stands for citric acid cycle. Excuse me. So apparently, exercise physiologists got together one time and said, "Hey, let's confuse everybody and call it 3 different things." No, that's not how it is. Normally there's scientific names behind them, and Krebs would be the individual who helped find it, basically discovered it. And then with this, the electron transport chain. The electron transport chain is the really important part with this, because this is where we're going to get a lot of the ATP, and this is where we're going to use the oxygen. So again, oxygen is required for it. VO2 max, and we'll talk more about VO2 max later on. But VO2 max is very important in aerobic fitness and determining your aerobic fitness. There's ways of estimating it. There's ways of getting a really, really accurate number. But VO2 max basically stands for the maximal amount of oxygen that you're able to utilize, that volume that you're able to utilize. And it's a really good indicator of how fit somebody is. Now, this is going to be highly efficient for ATP generation. You get about approximately 33 ATP per glucose molecule. So this is really efficient. You're providing a lot of ATP. Now, the important factors for that volume of oxygen that you're able to consume, thus how well the oxidative system is working and how fit you are. CVS, and that's not CVS the store, but that's CVS cardiovascular system, how well it's able to deliver the blood. And within the blood, we obviously have oxygen that is needed to be carried around through the blood. So how well the cardiovascular system is working. Pulmonary ventilation, how well the lungs are doing what they're supposed to be doing. And with the pulmonary system, the cardiovascular system kind of go hand in hand. If one's not working well, if there's some type of disease or condition, it's going to hinder the entire system. Also, the oxygenation of the blood itself. Within the blood, we have what's known as hemoglobin. Hemoglobin is what carries the oxygen around, as well as the utilization of oxygen. And this has to do with the mitochondria. Mitochondria, again, is known as the powerhouse of the cell. This is where a lot of the cellular processes occurs. This is where all the good stuff occurs, really. And within that mitochondria, there are enzymes known as oxidative enzymes. The enzymes that are associated with these systems, the Krebs cycle and the electron transport chain. So very important for mitochondria. In fact, when we get into shape, increasing our aerobic fitness, we increase those enzymes because we end up increasing the number and also a little bit of the size of each of those mitochondria. All right, so the oxidative system starts with glycolysis. So the ATP yield for that glycolysis is going to be the same. It's going to be the same steps, except that last step. We're not going to convert the pyruvate into lactate. We're just going to go right into the citric acid cycle. We're going to start off with converting pyruvate into what's known as acetyl-CoA. So again, in this case, oxygen is present. So this reaction is going to go this way instead of going into lactate. Now, the oxidative system is different. The big difference here is we continue on then. If we look at this figure, we see that glycolysis occurred in the cytosol. So this is not in the mitochondria of that cell. Once we get past pyruvate, we go into the mitochondria, and this is where the citric acid cycle was occurring. So again, remember from this point, we have 2 pyruvate. So there's 2 pyruvate here, and when it's converted into acetyl-CoA, it's just not one of them is converted to acetyl-CoA, 2 of them are. So we actually go through this cycle twice. Now, we also create additional ATP in one of the steps here about the 6:30, 7 o'clock position of the citric acid cycle. The whole point of the citric acid cycle is to create and transfer, I don't want to say create, to transfer basically hydrogen or electrons. All right, so we're starting off with either NAD or FAD. And through some of these steps, we actually are going to create either NADH or FADH2. Now that's going to be transported into the electron transport chain. And through a bunch of different reactions, then we're going to get the ATP. And if we see over in the electron transport chain, this is where we're adding oxygen. And that's why the oxygen is necessary. Now for this system, isocitrate dehydrogenase is the rate-limiting enzyme. Again, the more ATP that we have, it's going to decrease the activity of isocitrate dehydrogenase. The less ATP that we have, the more activity that's going to be associated with isocitrate dehydrogenase. So this is a figure from the book. The previous figure was not from the book. It was more of an overall general aspect of it. So again, we're in the mitochondria and we're going around the citric acid cycle. You can see the intermediates in there. And you can see the places where we have these electrons that are going to be transported. See the dotted line? They're going to be transported into the electron transport chain. And with the addition of oxygen, we're going to create some ATP. So this figure is just to show you where the isocitrate dehydrogenase is used, and it is used in this step right here, about the 3 o'clock position, going from isocitrate to alpha ketoglutarate. Again, you don't need to know these intermediates, just know the rate-limiting enzyme and why it's important. So the electron transport chain, and we're definitely not going to get into the nitty-gritty details of the electron transport chain. If you want to look it up, you can, but it can be a little mind-boggling sometimes to even go through it. But really what's happening is there's hydrogen carried. That's carried by NADH and FADH2, basically the electrons. And from that, we're going to be adding some oxygen. And we're going to end up with water and ATP. So you can see the amount of ATP for every NADH and the amount of ATP for FADH, and 2.5 versus 1.5 respectively. Now, one thing I did want to point out here, going back one slide, is in the details of the citric acid cycle, is you can see where carbon dioxide is being produced in this step as well as this step. And you can see even from the conversion from pyruvate to acetyl-CoA, we're getting a carbon dioxide as well. That's where the carbon dioxide is being generated when we exercise. And hopefully, if everything's working properly, we can transport that back to the lungs, get it to the lungs, get it back to the heart, get it back to the lungs, and then exhale it. All right, that's why the carbon dioxide starts to build up, is because of this aerobic system. All right, so this just demonstrates how much ATP is being generated through the aerobic system. So you don't have to know the details of every one of these. The important part here is the total energy yield. Approximately 32. And this 32 is starting off with glucose. If we happen to have started off with glycogen, it's going to be 33, because remember, when we start off with glucose, we end up using one additional ATP at the beginning, so we end up losing one. So anywhere from 32 to 33, depending on if we use glucose or glycogen. Now, in the book, as well as any other exercise physiology textbook, they will generally have a spot in the chapter of bioenergetics talking about this number. And this number will change depending on what source you're looking at. In fact, there are some sources that say anywhere from 36 to 39 ATP, just for the simple fact that we end up using about 4 or 5 ATP just to transport some of these things across the membrane of the mitochondria. So we end up losing some ATP just because we need some facilitated transport mechanism, and we end up using ATP for that. But really, anywhere in the mid 30s, if you want to just kind of have an average, basically in the mid 30s, 32 to 33 is usually what is accepted. All right, so we're looking at beta oxidation now. Beta oxidation, this is relating to fat. If we look at free fatty acids, we're going to be making that into acetyl-CoA. And remember, acetyl-CoA is basically the first step, the first intermediate there of the citric acid cycle. We're leading into the citric acid cycle. Now, it requires a couple ATP, but in the end, we end up getting a bunch of it. And if you can see here, the ATP yield, 129 ATP for every, or at least for this molecule. It's going to depend on how big that fatty acid really is, how many carbons are present, of how much ATP we're actually going to get. But in terms of this acid, palmitic acid, this fat, we're going to get about 129 ATP. So those couple we use at the beginning to break it down and get into the citric acid cycle really doesn't mean a whole lot. Now, the one thing to keep in mind, is that fat, although it provides a lot of energy, it's not as efficient, at least aerobically efficient, as carbohydrates. For every oxygen molecule, we get about 5.6 ATP for fat versus 6.3 ATP for carbohydrates. Another way of saying that is that it takes less oxygen to get as much ATP out of carbohydrates versus fats. Or if you want to look at it another way, it takes more oxygen to get the same amount of ATP out of fat as it does carbohydrates, excuse me. So carbohydrates are more efficient at providing energy. We'll see some ways later on as we become more in shape, our fitness level increases. We're actually going to prefer, at least at lower intensity exercises and at even relative exercise intensities before we became aerobically fit, that we end up starting to prefer fat and we start getting away from burning the carbohydrates because we're able to use the oxygen more efficiently. But we'll get into that later on. So again, carbohydrates are preferred, especially for high intensity exercise. Doesn't matter whether you're fit or unfit. At high intensity exercise, and especially at maximal exercise, you're going to be using carbohydrates. That's really all you're going to be using. Protein is rarely used as a substrate. In times of starvation, it can be used. Some amino acids can be converted into glucose. They can also be converted into some of the intermediates of the citric acid cycle, as well as glycolysis, pyruvate, acetyl-CoA. Now, again, I referred to this a little bit before, but the energy yield is actually a little bit higher than what is represented usually. It's usually about 5.5 kcals per gram, but we need to excrete that nitrogen. So we end up using up some ATP, which brings the yield down to about what it is for glucose or carbohydrates. So it's not as efficient. And in terms of your diet, approximately anywhere from 5 to 15% of your total caloric intake should come from protein, because it's not going to provide a huge source of energy stores at all, really. And really throughout the day, only about 5 to 10% of your total energy that you're using throughout the day is going to be coming from protein. So there's not a lot there that's actually broken down. Now, there's going to be some instances where it could be a little higher and whatnot, but generally, if you're well fed, not much of your energy is going to be coming from the breakdown of protein. So we can see here from the figure from the book, there are a couple different ways we can get energy. So we have the triglycerides in the adipose tissue. So we get fatty acids and going into acetyl-CoA. Now this, before I go any further, I do want to touch on these ketone bodies. These ketone bodies are actually what provides the fuel for the brain in the absence of carbohydrates. So if we run out of carbohydrates, this is what happens: is we end up breaking down some of the fats, or we could even bring it in from amino acids, bring it into acetyl-CoA, and then into what's known as ketone bodies. Those ketone bodies are able to be used by the brain for the activity that is needed in the brain. So the brain doesn't necessarily need glucose, but these ketone bodies are basically a skeleton form of glucose. So, but it's not as efficient. So carbohydrates are much better. Glucose is much better for the brain. So glycogen is not only in the liver. This is it is a little bit strange that the authors or whoever made this figure didn't put a muscle tissue up here either, because glycogen is also stored in the muscles, but it is stored as well in the liver. So then it's broken down and we go through glycolysis and we go through the citric acid cycle. And for the citric acid cycle, we are going to go through the electron transport chain as long as there's oxygen available. Now, in the case of protein, so we have a muscle tissue over here. We're going to break that down into amino acids. We're going to lose a little bit of ATP, getting rid of that nitrogen through the urea cycle, and we're going to excrete it through the urine. Now, the amino acids, certain amino acids can be created into intermediates of this Krebs cycle or to pyruvate or acetyl-CoA. So just depending on what the amino acid is and how it can be converted. So we can get energy from protein, but it's not as efficient. It's good to that we can do that in times when we need it, but you should not be relying on protein as your main energy source, especially during physical activity. All right. One thing that's important to understand with these energy, there is no on and off button. All three of them are always working. They're always providing some amount of ATP. It just matters which one is more predominant in providing the ATP. Now, there can be some exceptions if there's a transition period where one of the systems is not able to provide as much ATP, where the other one is just starting, but it's providing almost the same amount. So maybe that 20 to 30 second range, the ATP PCR system is starting to run out of providing enough ATP, but then the anaerobic glycolysis or glycolytic system is providing a good portion of it as well. So if we look at a 100-meter sprint, so not going to last very long depending on the individual, anywhere from 10 seconds to 15 seconds. Again, it's not very long. The ATP PCR system is going to dominate the ATP production. Now, the glycolytic and oxidative systems are going to provide some ATP. They're just not going to be as much, nearly as much as what the ATP PCR system is. If we look at a 10,000 meter run, or if we look at a marathon or a long distance cycling event, the oxidative system is going to be the one that's predominantly providing the ATP necessary for that activity. However, the ATP PCR and the glycolytic systems, the two anaerobic systems, are still providing some amount of ATP. They're just not providing a large amount of it. They're not predominantly providing the ATP necessary. So, and this is what I was talking about with the systems and how much they provide. Now, if we look at the rate of ATP generation, the PCR system provides a very high amount of ATP really quickly. Doesn't provide an enormous amount of it, as you can see in the bottom. And the same thing with glycolysis. It doesn't provide a large amount, but it does provide it fairly quickly. Now, with carbohydrate oxidation, which would be basically going through the Krebs cycle, the electron transport chain, it provides it at a decent rate, but it provides also a lot of it. And the same thing with fat stores. Fat oxidation doesn't provide a whole lot really quickly, but overall, in general, it provides a lot. And how much it can provide is really just dependent on how much extra fat you have on your body. Really. Now, some of the factors affecting the oxidative capacity of the body, if you increase the intensity of the exercise, you're increasing the ATP demand. You need more ATP to keep on going, especially to keep on going with that intensity. So you need to increase the rate of oxidative ATP that you're creating through the Krebs cycle, through all these systems. So if you're creating more ATP oxidatively, you need to increase the oxygen intake at the lungs. So therefore, our breathing increases, not only our rate of breathing, but each breath, the volume gets larger as well. We're going to also increase the oxygen delivery. In this case, the heart rate's going to go up. So oxygen levels entering and leaving the lungs is actually close to what it is at the muscles. So that's why it's really good to measure if we go into a lab, and you can look this up on YouTube. There are plenty of videos looking at a metabolic cart to measure the amount of oxygen that you're using, and we're really measuring that at the lungs. But what's happening at the lungs is a good representation of what's happening at the cells. And there's also a very, very small amount of oxygen that's actually stored. So any calculations that we might do in a lab setting, there's going to be a very, very minute error because there's going to be some stored oxygen, but it's so small, it's really not going to be really a problem in any calculations that happens. And this oxygen, we're either going to use it or we're going to lose it. We're going to use it for something else, whatever we do. All right, so this is the percentages of either anaerobic or aerobic systems, and there's different events, different activities that we're performing here. So let's look at weightlifting. For the aerobic system, 0%, it says 0% is coming from the aerobic system. One thing I do not like about this figure is that it is not 0%. Now, you can make the case that within the first couple seconds, there's really not, it's probably less than 1% that's coming from the aerobic system, but the aerobic system is still working. It's still on. There is no on and off button. And the same thing on the other extreme, this jogging or 10,000 meter run, it's not 0% anaerobic. All right. So it's probably less than 1%, but generally speaking, you're, at least if I were making a figure like this, I would just put 1%. Right, or maybe 0.5%, or put something, or maybe just put less than 1%, probably would have been better. So, but if we go to a sport like soccer or field hockey, right, 30% of it's coming from aerobic and 70% coming from anaerobic. And if you think about sports like field hockey and soccer, there's a lot of movement, and you can have times where you're moving, where those athletes are continuously moving for a few minutes at a time without much stoppage. Or you could have periods where they basically sprint for a little bit, maybe for 15, 20 seconds, and then they'll stop or slow down. And then they'll speed up again, and then they'll slow down or stop, and then they'll speed up again. And then they might go for another 2 minutes without stopping. So sports like that, basketball is kind of the same way sometimes. But once you get into running, especially track events, that's more easily defined how much you're running and how much could be provided from each of the energy stores. So, all right, so that was chapter 3, Bioenergetics. And if you have any questions about the chapter, anything that I went over, please feel free to email me. Or even put comments in the comment section here in this video, if you would like, or on Blackboard. So the next chapter is going to be chapter 4, exercise metabolism. And I thank you for watching. Thank you for listening. Again, if you have any questions, please let me know.